Amitadi, we are starting.
Okay, start Karan.
Okay, wishing good morning to all participants. I, Dr. Paramita Mojumdar, on behalf of Department of Chemistry, Bethune College, welcome you all to today's webinar, Biomimetics Challenge to Chemists. This webinar is organized by Department of Chemistry, Bethune College under DBT Star College Scheme. This is the first such webinar organized by the department under DBT Star College Scheme. In today's webinar, we will have an opportunity to listen to two very eminent scientists of our country, Professor G. Mugesh from IISc Bangalore and Professor T.K. Pine from IISc Kolkata. But before we move on to the lectures, I would like to request my departmental senior colleague and coordinator of DBT Star College Scheme, Dr. Amita Kaur, to please deliver the welcome address for this webinar. And before Amitadi starts, I would also like to request all participants to please disable your audio and video so that the webinar can run smoothly. Amitadi, Dr. Amita Kaur, if you please deliver the welcome address.
Thank you, Paramita. It's very unfortunate for us that our principal, ma'am, Professor Krishna Roy, could not be with us today. She has just had an eye operation, and we all wish her a speedy recovery. But on behalf of all, and also on behalf of our principal, ma'am, I welcome you all to the one-day webinar entitled Biomimetics Challenge to Chemists, organized by the Department of Chemistry, Bethune College, under the DBT Star College scheme. I take this opportunity, obviously, to welcome to extend my warm and hearty welcome to our two very distinguished speakers, Professor G. Mugesh of IISc Bangalore and Professor T.K. Pine of IACS Kolkata. I also extend my welcome to all the participants, especially I would mention my colleagues, my teachers, my students, the scholars, and all the others who are participating. Because the situation is very unusual, so obviously we cannot meet in person, so we have resorted to this virtual platform. Now this webinar is organized under the DBT Star College scheme. DBT Star College scheme is actually a project that was launched by the Department of Biotechnology, Government of India. Its main objective or aim was to attract students and encourage them to pursue careers and take up studies in basic sciences at the undergraduate level. With the years, we have seen that students are not very interested in studying basic sciences at the undergraduate level. So automatically, this is mainly because the laboratories in the different colleges are not well equipped. So under the DBT Star College scheme, the project obviously provides financial assistance to colleges which have undergraduate courses in basic sciences to obviously convert their laboratories to well-equipped laboratories through the purchase of equipments, instruments, and other consumables. It also provides financial assistance for the faculty development programs. And by this, the faculties can upgrade their knowledge with the changing time. It also provides financial assistance to enhance the skills of the skilled laboratory workers and automatically help us out the laboratory assistants. And with this, obviously, the laboratory becomes more well equipped. It provides financial assistance for organizing seminars, workshop, projects, for visits to different academic institutions, to different industries. So the students can obviously become more knowledgeable about what is happening around them, and they become involved in the experimental skills. It actually opens the students and allows them to explore the experimental world. And in this way, the students are motivated and they become more independent, more logical, and they can obviously cultivate their capabilities and obviously their creativities. And as I've already mentioned, because of the pandemic situation, we could not organize a seminar. So we have resorted to this virtual platform of organizing and webinar. And on this webinar deals with biomimetics, challenge to chemists. Biomimetics literally means, biomimetics or biomimicry also it is known as, literally means imitation of the living. It is the emulation of the models, the systems, and elements of nature for the purpose of solving complex human problems. According to Biomimicry Institute, it can be defined as an approach to innovation that seeks sustainable solutions to human challenges by emulating nature's time tested patterns and strategies. The goal is to create processes and policies, new ways of living that are well adapted to life on Earth over the long hour. Central idea is nature has already fixed many problems that the society faces. Animals, plants, microorganisms are experienced engineers. They now they know what works, what is appropriate, and what lasts. The main belief of biomimetic approach is what is around us is the secret to survival. Thus, the designs following the biomimetics will ultimately allow human products to be more efficient, resilient, and sustainable. Now, we can find examples of biomimetics or biomimicry. The first flying machine, heavier than air, designed by the Wright brothers in 1903, was inspired by flying pigeons. Then we can talk of obviously the climbing pads. They are a mimic of the biomechanics of the gecko. Then the velcros, which is very common now, is born from the study of hooks implemented by some plants for propagation of seeds via animal coats. It finds application more or less indirectly to all sections of life. Biomimetic chemistry deals mainly with chemical systems that can simulate biological phenomena, particularly catalysis and specificity, example, creation of artificial enzymes and life-saving drugs. And the earliest example of such was the work of Sir Robert Robinson, that is the synthesis of the alkaloid troponone. And later, E.J. Corey also has been working on carbenium-mediated cyclization of linear polyenes to a tetracyclic steroidal ring systems. And now, obviously, the work is going on in the field of creation of artificial enzymes and the life-saving drugs. I am sure you all must be very eager and restless to hear more about this from our very well-esteemed speakers. So again, I welcome you all to this webinar, and I'm sure at the end of the day, we will all have the satisfaction of becoming more knowledgeable and enriched on the subject. Thank you. Over to Paramita.
Thank you, Amitadi. Now, it is time for us to move on to the first technical session of today's webinar. The first lecture will be delivered by Professor G. Mugesh from IISc Bangalore. Professor Mugesh completed his graduate studies in chemistry from University of Madras and then obtained master's degree from Bharathidasan University. He then completed his PhD from IIT Mumbai. There he continued his postdoctoral studies till 2000, and then he moved for his further postdoctoral work in D. Wolf Walther D. Mont of Brunswick University of Technology and D. Helmut Sees of University Dusseldorf on Alexander von Humboldt Fellowship. Then he moved to Scripps Research Institute with Skaggs postdoctoral fellowship. In 2002, Professor Mugesh joined IISc. Professor Mugesh received a number of national and international awards. Only to mention a few, he was awarded award for young chemists by IUPAC and Alexander von Humboldt Foundation Equipment Award in 2005. Young affiliate by the World Academy of Sciences in 2008. Yuki Eri Standard Research Award in 2009. Bronze Medal of Chemical Research Society of India, Young Scientist Award of Inter Academy Panel, RSC West India Young Scientist Award, and Award for Excellence in Drug Research of Central Drug Research Institute, all in 2010. In 2011, he was chosen for AstraZeneca Excellence in Chemical Award. He was awarded with Shanti Swarup Bhatnagar Prize by CSIR in 2012. In 2013, Federation of Asian Chemical Societies awarded him with Rising Star Commemorative Plaque, ISCB Award for Excellence by Indian Society of Chemists and Biologists was given to Professor Mugesh in 2016. Along with all these and many other awards and fellowships, Professor Mugesh is also elected as a fellow by many science academies like Indian Academy of Science, National Academy of Sciences, India, Royal Society of Chemistry, and Indian National Science Academy. He has innumerable publications in the most leading international journals in all these years. His group and Professor Mugesh is still continuing their research in small molecule-based glutathione peroxidase mimetics, nanoenzymes for biomedical applications, chemistry of the thyroid hormone metabolism, halogen bond-mediated cell membrane transport, expanding the genetic code by unnatural amino acids, and such works. Sir, I would request you to please enrich us with today's lecture. We are waiting, our students and ourselves are eagerly waiting for your talk. Professor Mugesh.
Yeah, thank you. Good morning. Thank you, Dr. Mazumdar, for the nice introduction. I'm happy to present this talk. As mentioned earlier, it's unusual situation, and I would have liked to come personally and deliver this talk, but unfortunately, this is not possible now. I thank the organizers, the Bethune College, for inviting me to give this talk. So I'm going to talk about artificial enzymes. I'll share my slides. I hope you are able to see the slides.
Yes, sir. We can see the slide.
So next 35-40 minutes or so, I'm going to talk about artificial enzymes and how one can use artificial enzymes to regulate biological systems. Okay, so when you talk about enzymes, and we all know about enzymes, right? The biomimetic systems, there are multiple ways one can mimic the biological functions, biological systems. And someone designed, for example, in Japan, people trained, design trains based on bird's beak, right? The Shinkansen, the bullet trains. And some people design certain polymers taking idea from the nature. Our work is mainly, taking enzymes, understanding enzymes, and designing small molecules that functionally mimic the function of the enzymes. So Enzymes are biomolecules. I'm sure I don't need to give an introduction to enzymes because all of you know what are enzymes, right? They are biological molecules, typically proteins. There are non-proteins as well. And these enzymes speed up chemical reactions that support life, right? And that's the reason we call them biological catalysts. So they play essential role in any of the biological function you take, whether you take respiration, digestion of food, muscle and nerve function, or many other functions, right? So they bind to molecules and alter them in very specific way, right? The human body contains hundreds of such enzymes. For example, amylase that converts starch into sugars. Okay, so this enzyme is present in saliva that begins the chemical process of digestion. Okay, you can see the food is broken down to starch, glucose, maltose, and glucose finally. So this is the most important source of energy in all organisms. Okay, so this is one type of enzyme, amylase. There is another type of enzymes which are important for nerve functions. For example, acetylcholine esterase is an enzyme which is involved in the breakdown of acetylcholine. You can see acetylcholine, which is a nerve transmitter, and breaking down acetylcholine to acetate and choline controls the nerve function. Okay, and this is an enzymatic reaction. Okay, and this is important to control the brain function. This is another enzyme, the key enzyme that we know. And there are enzymes involved in making DNA, for example, DNA polymerase. Okay, polymerizing DNA, getting multiple copies of DNA. This is another type of enzymes, DNA polymerase, which is involved in polymerization of the genetic materials. So, what are artificial enzymes? When we talk about enzymes, we know they are very well structured. The active site is developed in a proper way and they are produced in response to biological stimuli and they are evolved. That means the nature optimized the conditions over hundreds of years to evolve system that perfectly function in human body, right? But when you would like to mimic that, lot of efforts are required, right? That is the challenge to the chemists. How do you design, how do you modify an enzyme, a synthetic compound to function as enzymes? So this is a very simple concept. We know there are enzymes having active site, the substrate comes and bind to the active site, then the enzyme substrate complex is formed, then the reaction takes place at the active site and then the products are eliminated, right? This is what we know, simple enzymatic reactions. So I have shown one active site, which is a heme active site in metalloproteins. So a simple chemical catalyst, if that can be achieved to mimic some desired function or features of the enzymes, so we call them artificial enzymes, right? And a new area is emerging as synthetic biology, which talks about synthesizing biological systems. Okay. So in 2015, catalyst from synthetic genetic polymer has been achieved. However, it is challenging to design artificial enzymes that can perform specific reactions inside the living cells because most of the biomimetic enzymes or artificial enzymes function outside. Okay, there is no problem. You can understand the active site, design a compound, and then that function as an enzyme. But does it work inside the cells like enzymes? Okay, because there are multiple things need to be taken into account while designing a compound that can function inside the enzyme without generating any unwanted reactions. So my group in this talk, I'm going to talk about something very general, but also a little bit about what we are doing may be useful for some of the students. The aim of our study is to design artificial enzymes to regulate metabolic, cardiovascular, and neurological processes under disease condition, because that is important, right? So when somebody is healthy, then we don't need to design an artificial enzyme, right? So when disease occurs, when somebody is under, has a disease, and the natural enzymes do not function well, that is where the artificial enzymes will be useful, right? So before going to the talk, because I'm going to talk about some of the role of metals, as well as role of the active site, designing active site based on enzymes. The general question that we ask is life organic, right? If you look at Earth's crust, you can see oxygen is the maximum 46.4%, silicon, and then aluminum, iron, calcium, sodium, magnesium, and so on. But if you look at the human body, okay, oxygen is of course 61%, carbon 23%, hydrogen 10%, nitrogen 2.6%, calcium 1.4%, phosphorus 1.1%. So if you consider these major elements, carbon, oxygen, hydrogen, nitrogen, phosphorus, sulfur, which are considered as organic molecules, right? When they are part of organic molecules, that constitute 98% of the human body. For example, carbohydrates, nucleic acids, proteins, hormones, vitamins, right? But what about the metal? You compare, for example, iron in seawater, 0.005, and in human plasma. If you take the ratio of these two, the iron in human plasma is 400,000 higher than that of seawater. Zinc is 215 times, copper 1650 times, molybdenum 100 times. So what does it mean? This means that metal ions also play important role, and although in very small amount. Okay, so we have been studying, the role of such metal ions. So when you talk about metal ions, people say that they can be toxic. Okay, when you do a chemical reaction outside the human body, there is no problem. But if you take it inside the human body, they may become toxic. Okay, so, but all things can be poisons, right? I'm sure you all agree with this statement. Anything you take excess is going to be poison, right? Even food, if you take excess, it's going to be poison, right? So then when you increase the concentration, the growth increases, then there is a slow drop in the health. Okay, so that's due to excess amount of these species, these metals, or whatever the compound you take. So it all depends on the dosage, individual health, and way of administration. So we have been working on a single atom change in biology. So how a single atom change affected the biological system? Can we use that to mimic the enzymes to design artificial enzymes? Okay. So you may know, some of you know, that approximately 28 elements are considered as the elements of life. Okay. So that means they are essential for human life. Many of them are main group elements, and most of the first row transition metals. But one atom change, for example, zinc is essential element. Without zinc, we cannot survive. But the next two elements, cadmium and mercury, are toxic, right? So if you take group 16 element, oxygen, sulfur, very important, essential. Selenium is essential, but when you go to tellurium, it becomes toxic. You go to the halogens, the nature avoided fluorine, chlorine, bromine, particularly in human systems, but it chose iodine, right? Leaving the lighter elements, going for a heavier. There must be a reason for that, right? So in today's talk, I will tell you why is it important to design artificial enzymes mimicking the biological systems to control the oxygen metabolism. Okay, hydrogen peroxide and the central redox theory for aerobic life. Okay, and you all know that oxygen is important, right? Nobody can deny that. And we breathe oxygen, and respiration, 95% is used in respiration. It's important, essential, without which we cannot survive. But what happens when you breathe? What is happening to the oxygen? When oxygen goes to all the cells, what is really happening to the molecular oxygen? So the oxygen is converted to water, right? That is what happens in human body. Oxygen is converted to water. But in this process, it can generate a variety of uh, oxygen containing species. If it undergo one electron reduction, it produces superoxide. Two electron, add one more electron, hydrogen peroxide. One more electron, hydroxyl radical. Fourth electron gives you water. So the human body has a number of enzymes that controls this level: superoxide, hydrogen peroxide, hydroxyl radicals. Okay, when you look at hydrogen peroxide, I put this picture. If you open the hydrogen peroxide with the bare hand, what you see? You see a white color, right? And that is because the skin proteins get oxidized. If that is the case, when we have hydrogen peroxide inside our human body, how it is controlled? Okay, so these are controlled by enzymes like superoxide dismutase, which takes care of the superoxide, glutathione peroxidase, and catalase. These take care of the hydrogen peroxide. Okay, so these species, superoxide, hydrogen peroxide, and hydroxyl radical, are called partially reduced oxygen species because it's not a complete reduction. So if it is a complete reduction, it should go to water. But they are only reduced by 1 electron, 2 electron, 3 electrons. Okay, they are partially reduced oxygen species or reactive oxygen species. And then if there is an imbalance between the reactive oxygen species generating system, these are the generating system, and the scavenging system, that lead to a redox imbalance and oxidative stress. Okay, so that's the reason I will ask you a simple question. Okay, when we put iron chain or iron rod outside, right, you have this one. So after few days, what you see is this one, right? The color changes and it becomes like this because we call it as rusting, right? Iron rusting. So when iron, water, and oxygen react together, rusting takes place. Okay, but the question is, human body has about 4 to 5 grams of iron, and only 1 milligram is lost per day. And why no rusting takes place inside? If rusting takes place inside, it lead to diseases, right? Neurological diseases, Alzheimer disease, Parkinson disease is nothing but rusting, right? It's basically dysregulation of iron, dysregulation of metal ions in the brain. That's the reason the iron in biology is tightly regulated, because it cannot afford to lose the iron. You know, the human body cannot just allow the iron to be floating around in the plasma or the blood, right? Because it's going to lead to rusting and it lead to diseases. That's the reason there are enzyme systems that exist to control the reactive oxygen species reacting with the free metal ions. Okay, because this can lead to hydroxyl radical, DNA damage, lipid oxidation, and so on. So peroxyredoxins, glutathione peroxidase, catalase, so three key enzymes that control hydrogen peroxide. Why hydrogen peroxide? Hydrogen peroxide is a necessary evil for cell signaling, which is important. Without hydrogen peroxide, the redox signaling does not start. So the protein thiol should get oxidized to protein sulfonic acid to initiate the redox signaling. Okay, so that's why this is called signaling and this is called oxidative stress. So distinguishing these two is really a challenge. So designing an artificial enzymes to control such species in a proper way is going to be a challenging issue. Okay, so I will pick up one or two examples to tell you how we go about it. Okay, so first you choose enzyme, which enzyme you want to function, and we chose the glutathione peroxidase because glutathione peroxidase is the one that controls the hydrogen peroxide and organic peroxide levels. You can see that the organic peroxide is converted to alcohol or hydrogen peroxide is converted to water by using glutathione peroxidase as an enzyme. So very unusual enzyme. Why I wanted to work on this is very unusual enzyme because this enzyme contains selenocysteine in the active site. And I'm sure many of you have not heard about it, right? Selenocysteine in a protein. And this is called 21st amino acid. We know 20 genetically encoded amino acids, right? And but selenocysteine is considered as the 21st amino acid. So it has amazing property in human body. Why nature shows selenocysteine? I was talking about single atom change, right? Oxygen, sulfur, selenium, tellurium. This enzyme catalytically reduce hydrogen peroxide to water without any partially reduced oxygen species, without any other reactive oxygen species. It is just a clean conversion of hydrogen peroxide to water. It's interesting, right? Using a biological thiol as cofactor, which is glutathione. So I want to first mimic this enzyme with the small molecules. As a chemist, I am interested because there are many disease conditions where the GPX is less expressed or inactivated in human body. So therefore, the function of this enzyme is affected in human body when the disease happens. So the fundamental idea is to mimic this enzyme so that you can use those enzyme mimetics to regulate such small molecules, biological functions. Okay. So first thing we would like to look at it is why selenocysteine, right? Why not cysteine? Why not serine? Why nature chose selenocysteine? Because the pKa of a thiol group in serine is 13 and the thiol pKa is 8.3 in cysteine and then 5.2 in selenocysteine. So the pKa, a drastic decrease in the pKa. That means that a selenocysteine in a protein is a powerful nucleophile. Okay, much more powerful than a cysteine and a serine nucleophiles. This is one primary reason why nature chose selenocysteine for these reactions. So sometime in 1980s, people started working on can we get a small molecule which functionally mimic this enzyme. So this is a huge protein. You know, it has a selenocysteine and multiple units. They interact with each other and so on. It has a catalytic triad like this. So if you want to mimic that enzyme, so first thing is you should have a selenium because the enzyme has a selenocysteine in the active site, right? So selenium should be there. So therefore, people made such a small molecule and it has undergone several clinical trials as antioxidant, anti-inflammatory and atherosclerotic and antithrombotic activities. Okay. So selenium deficiency is detrimental for normal growth and development in mammals. And they have a narrow range, if narrow range of chemopreventive, chemotherapeutic, but it goes to higher level, it become acutely toxic. Okay. So Brazil nuts has the highest amount of selenium. So people who have selenium deficiency are treated with the Brazil nuts, 83 microgram of selenium per grams of this nut. Huge amount of selenium present in these Brazil nuts. Okay. And this is the metabolic pathway of selenium because selenium is essential, which means one cannot survive without selenium in the human body. So therefore a metabolic pathway exists. I'm not going into the details of this. Okay, this is a small molecule reported in 1984 as an artificial enzyme, artificial small molecule which function as artificial enzyme glutathione peroxidase. So when you substitute the selenium with sulfur, it becomes inactive. So that means that nature purposely selected carefully the selenium because it's redox active, right? And our group extensively studied about the antioxidant and enzyme mimetic activity of epsilon. And the cyclic compound 5-membered ring is important because it undergo a reversible cyclization like a protein found in human body, that is a PTP1B, which undergo a reversible cyclization to protect the active site. And we found that it's amazingly similar, right? Except that you have a sulfur here and a selenium here. So in a number of studies, we showed that the reversible cyclization protected the selenium. Okay, so this is another aspect we look at it when we want to make artificial enzymes for natural systems. Okay, mimicking the biological systems. We look at it how the metabolism happens and how toxic these compounds are to mammalian cells. Okay, and fortunately this compound is non-toxic to human cells. Okay, that's the reason it is undergoing several clinical trials. So we prepared several such compounds. You can see that epsilon and its analogs, and diselenides having a methoxic group here. So many structural analogs we prepared and studied the enzyme mimetic activity. Okay, so what we found was that this compound, which is mimicking the glutathione peroxidase. Okay, this is an excellent mimic of glutathione peroxidase, and this can happen by rational design to improve the GPX activity of epsilon. This is where the chemistry comes into play, right? So it's not all molecules are similar, right? You can induce a function. So making molecules in the lab is not very difficult, right? You can make hundreds of molecules. Okay. So by linking atoms by atom, right, you can make molecules, making carbon-carbon bonds, making carbon heteroatom bond, carbon-nitrogen bonds, right? But what is challenging is making molecules with a purpose. Okay. We should not simply make molecules. So it's not that we can make molecules, we should make molecules. We should make molecules with a purpose. So there should be a molecular design, a fine balance of chemical reactivity, biological activity, and toxicity. You may have a chemical reactivity high in the lab if you do, but how biologically active they are and how toxic they are to human body. These are important if you want to design artificial enzymes, particularly for treating diseases. Okay, and we study the catalytic mechanisms using standard chemical methods, spectroscopic methods. You analyze the intermediates, you isolate them, you characterize them, then you propose a mechanism. This is how this molecule functions. Then you compare the function with the enzymes. Is it working like enzymes, or there is a difference in the hello?
Yes, sir. You can continue.
Okay. Yeah. So this is the catalytic mechanism that we study. Okay. So now the next step. Okay. So now you started with the small, very basic chemical synthesis. You got an idea from an enzyme. This is how the enzyme functions. This is the active site, and the active site is activated like this. This is the pK of the enzyme active site. And then in the test tube, you have done the experiment, or in the flask, you have done the experiments. It works well. It mimics the enzyme. But now the next step is, does it mimic the enzyme inside the cells, inside a human cell? Okay. So then you move from the idea that you got from the synthesis and in vitro studies to understand how these compounds function inside the cells. So So in these particular studies, again, I am not going into the details. This is the glutathione peroxidase mimetic we developed to control the hydrogen peroxide level in the human cells. As you can see here, these the 6 and 4A are our compound. 6 is epsilon and 4A is our compound. You can see the oxidative stress induced by hydrogen peroxide. This is a fluorescent probe that we use to detect the level of hydrogen peroxide inside the cells. So if you see that, the fluorescent enhancement due to hydrogen peroxide, oxidative stress is brought back to the normal level by treating this with this compound. So that means this compound functionally mimic or biomimic the glutathione peroxidase. Okay, it's working in the cells. So that means these cells take up these compounds and then perform a similar reaction. Okay, so the same reaction that an enzyme performs. This also prevents a DNA damage because, as I mentioned in my introductory slides, hydrogen peroxide can damage DNA. Right? You can see with hydrogen peroxide, a lot of DNA damage happens. It can break a single strand or a double strand. Double strand, repairing double strand break is much more difficult than repairing a single strand break. And when treating with our compound, you can see completely prevented the DNA damage. So that means these small molecule that we prepared in the lab control the hydrogen peroxide level like glutathione peroxidase enzyme and also prevent the DNA damage. This is what we are looking at it. Can we mimic the function inside the cells? So we also studied extensively about thiol exchange reactions, which you may not be so much interested in. Okay, these are all unexpected complications. So when we talk about, we come across unexpected complications, unexpected reactions. Okay, most of the time things don't work the way we wanted that to work, right? So we get failure and then we learn from our failure and then we take it forward. Research is all about that, right? If you get success every day, then every day you can make breakthroughs, right? But the research doesn't go in that way, right? You work years together uh, to understand something. But here, why I am bringing this, the connection between my previous part of my talk to the next few minutes. This compound was discovered as glutathione peroxidase enzyme mimetic in 1980s, right? And multiple clinical trials have been carried out, but so far this has not entered the market. It has not entered the clinic. Okay, because of such complicated reactions. Okay, it undergoes thiol exchange, not allowing this to functionally fully mimic the enzymes. Okay, it's active but not the desired function of the enzyme that you can achieve by using these small molecules. But recently, such unexpected complications people have used to use this compound for COVID-19. Okay, so this is the thiol exchange reactions. When you have a cysteine, it forms a disulfide bond, and another thiol comes and cleave the disulfide bond. New disulfide will be formed. This is called thiol exchange reactions. Okay, and these are all, for example, a protein where cysteine is the target, and also disulfide bond formation, which make the protein compact. It's involved in protein folding and so on. Okay, and recently this is what we have shown, that epsilon can irreversibly block an enzyme by forming a covalent selenium sulfur bond. It's a covalent inhibition, right? You're blocking an enzyme activity. In diseases, this is important. Many of the drugs that we use, they depend on inhibition of enzymes. So some enzymes are overactive, so you want to inhibit that enzyme with small molecules, inhibitors, and those inhibitors become drugs, right? And this one we have shown that epsilon can covalently react with a cysteine, irreversibly blocking that. And a similar idea is used recently. And the human trial suggests rescue drug could be safer treatment for bipolar disorder. Okay. And this was reported sometime in 2013. And epsilon has gone from identification as a potential bipolar treatment to human trial in about 2 years, incredibly fast for any drug discovery process, because epsilon is safe, right? It discovered in 1980s, but it's found to be safe to human. So if somebody can find an application for epsilon, they can be injected directly to human. It is part of the National Institute of Health clinical collection, right? So this one recently used as a drug. For example, targeting Mycobacterium tuberculosis, right? It's a bacteria, and epsilon block the reactive cysteine residues. Okay, this enzyme uses cysteine residues for activity. So by blocking this, by forming a covalent bond, is a way of inhibiting the enzyme, right? And this is the very recent one after the COVID breakdown. This was published in April 2020. This is the crystal structure of the SARS-CoV-2 main protease that provided basis for the design of improved inhibitors, right? And this is called MPRO. That means it's a main protease of COVID-19, one of the best characterized drug targets among coronaviruses. Okay, and this is essential. This enzyme is essential for processing the polyproteins that are translated from viral RNA. Okay, therefore, inhibiting the activity of the enzyme would block viral replication. So no human protease with a similar cleavage specificity are known. So therefore, the inhibitors are unlikely to be toxic to human body. And using this strategy, recently, uh, this group published a paper in Nature, structure of main protease from COVID-19 virus and discovery of inhibitors. What they did, they screened 10,000 compounds and identified this particular compound epsilon as one of the 6 potential COVID-19 therapeutics. So now this is undergoing many trials to understand whether epsilon can be used to treat viral infection like COVID-19. And this is how, a basic research, understanding a function of proteins and functionally mimicking the proteins with the small molecules, understanding the small molecules, the targets in human body, whether they target a specific proteins. And if you find that they can target a specific proteins, this can be used to inhibit those enzymes, proteins, which cause diseases, whether it is a human protein or it is a bacterial protein or viral, it doesn't matter. Okay, you can inhibit that. So this is the molecular characterization of this. Again, another paper appeared. Molecular characteristics of epsilon binding activity to COVID-19 main protease. Again, they talked about the epsilon, how it binds to the main protease. And this is interesting because epsilon, this is the targeting the viral protein, but this also controls the reactive oxygen species, because the COVID alters the human biology, human physiological processes, including it alteration in the reactive oxygen species, cytokines, neutrophils, okay, ferritin levels, interleukin 6, all these are involved, for example, in pulmonary inflammation, lung injury, thrombosis, vascular inflammation. So such compounds may be used for viral replication, but at the same time takes care of the damages that is caused by the viral infections. Okay, so in today's talk, I talked about how you can design small molecule that can functionally mimic enzymes. Okay, only one example I picked up. We work on several other enzymes and a lot of other activities which I didn't have time to present. But one example I picked up and I showed how you can proceed, how the chemistry can help in understanding this and understanding a little bit biology later to take this to the biological systems. So these are the students involved. This is my group, which is a multidisciplinary group. When we study chemistry, we are more restricted ourselves to compartmentalization, right? Inorganic chemistry, organic chemistry, physical chemistry, theoretical chemistry and so on. But when you come to research, if you restrict yourself like that, you will not be able to achieve anything big which is useful to the human society. So you need to keep your options open. You need to understand other areas, understand different disciplines, bring people together to work on those areas, and take a big picture in mind. What is that you want to achieve? Okay, not publishing papers alone, right? What is the problem that you are trying to address? That is more important than which discipline you are working on. Okay, this breaking the boundaries, that is important, right? Move beyond the conventional areas, move beyond the narrow areas of research. Okay, try to understand the big picture and take up problems like that. So that's the uh, suggestion I would like to have. I would like to tell all the students. Thank you very much.
Thank you, sir. Thank you very much. It was such an informative and intriguing lecture. Sir, I would like to request you to answer a few questions. There are
Sure,
probably three questions.
sure.
The first one says that does selenocysteine have a corresponding codon, or is it formed by post-translational modification, maybe adding selenium to a cysteine?
Yeah, it's a very interesting question. Uh, there is no specific codon for selenocysteine. It uses UGA codon, which is a stop codon. Okay. It is not a post-translational modification. It happens co-translationally, and the modifications happens on tRNA. Okay. So where a serine residue is changed to selenocysteine. So nowhere cysteine is used. It's a serine which is changed to selenocysteine, but it doesn't happen on the protein. It happens on the tRNA when serine is attached to the tRNA. So this involves several selenocysteine recognizing elements. Okay, there is a selenophosphate synthetase which introduces selenium to the serine and use UGA as the codon, which is a stop codon. Usually the protein synthesis stops when the UGA codon comes in, right, during the translation on ribosomes. But here what happens is it continues the protein synthesis incorporating selenocysteine instead of uh, cysteine in that place.
Okay, sir. Thank you. The second question is how nucleophilicity increase helps in converting from water from H2O to water in context of selenocysteine.
Yeah, selenocysteine is a nucleophile, so it attacks at the hydrogen peroxide. It's a nucleophilic attack at hydrogen peroxide. Then it forms a selenic acid because hydrogen peroxide gets reduced to water and the selenium selenolate, which is in the reduced form, gets oxidized to selenic acid, SeOH, which is similar to the sulfenic acid, SOH in biological systems. So the selenocysteine get easily oxidized to selenic acid much more rapidly than cysteine. So that's the reason why it's very active.
Okay, sir. The last question is flexizymes have been known to expand the genetic code to unnatural amino acids. However, do you think that incorporation of such flexizymes in biological systems would have any adverse effect on the canonical protein machinery already present?
Yeah, it's not, people are working on incorporation of unnatural amino acids by modifying, enzymes, for example, tRNA synthetase. Basically you are expanding the genetic code with unnatural amino acids. And at the protein levels, when you are making a protein with unnatural amino acids, it's not going to affect the natural translational machinery because this is orthogonal to the host system. Okay, whatever we are working on, for example, incorporation of unnatural amino acid using orthogonal technology. So that means it doesn't interfere with the host system. So it is completely a new pathway that is being used, but it uses all the machineries of the host systems, but not the translation machinery. It doesn't interfere with the translation machinery.
Okay, sir. So these were the questions. Thank you, sir. Thank you very much for such a wonderful talk. I think all of us have highly benefited from your talk. Thank you, sir.
Thank you very much.
Okay, so now this is the time for us to move on to the second talk. And the second talk will be delivered by Professor T.K. Pine from IACS Kolkata. Professor Pine completed his MSc from IIT Kharagpur. He did his PhD from Max Planck Institute for Bioinorganic Chemistry in Germany. And then he did his postdoctoral research from Department of Chemistry and Center for Metals in Biocatalysis, University of Minnesota, USA. Professor Pine joined Department of Inorganic Chemistry, ICS Kolkata, as a visiting faculty in 2006, where he is still continuing as an eminent scientist. Professor Pine was awarded INSA Medal for Young Scientist in 2011, A.V. Ramarao Foundation Young Scientist Award in 2012, Professor B.N. Ghosh 80th Birth Commemoration Award by Indian Chemical Society in 2018, and Chemical Research Society of India has bestowed Bronze Medal to Professor Pine in 2019. Professor Pine has also been Editorial Advisory Board member of Journal of Biological Inorganic Chemistry and Inorganic Chemistry Journal. Sir, I would like to request you to please deliver your talk today. Professor Pine.
মানে উনি কি জয়েন করেছেন পারমিতা? উনি আমায় বললেন জয়েন করছেন এক্ষুনি।
আচ্ছা, okay, so if he has not joined, so just let's wait for one more minute. Probably he will join.
উনি জয়েন করছেন। আকাশ বাবু, আপনি ওনাকে জয়েন করতে দিবেন কিন্তু।
Yes, it's open. He hasn't come yet.
আমাকে তো বললেন জয়েন করে যাচ্ছেন।
He has joined.
এখানে তো জয়েন করেছে দেখাচ্ছেন। ঠিক আছে, তো জয়েন করে গেছেন।
আচ্ছা, জয়েন করেছেন, ঠিক আছে। পারমিতা, তাহলে তুমি ওনাকে বলে দাও। ঠিক আছে।
Professor T.K. Pine. Professor Pine.
Yeah, can you hear me?
Hear us?
Yes, we can hear you, sir.
Yes, 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 sir. Yes, sir. So please. Yes.
Oh, it's already my time?
Yes, it's your time, sir.
Yes,
okay.
Sir.
All right. So thank you very much. Thank you very much for having me here. And it's really a very difficult situation that all of we are going through. And I know we cannot see the face of the people who are in the audience, yet this is what the life is. So we have to go like this. So let me share with you my presentation slide for today. Okay. All right. So can you see my presentation slide?
Yes, sir, we can see.
Yes.
Okay. Okay. Thank you very much. All right. So considering the presence of a large number of undergraduate students here in the audience, I thought of take today's story from the very basics. Then slowly I'll take up the story, and then at the end I'll talk about some of the research activities that is being carried out in my research group here at ISES. And I'll just take you through the way we understand this particular topic. So here it goes. The title of today's talk is the reductive activation of dioxygen by iron-containing enzymes, and what mechanistic information we get from synthetic chemistry, particularly from biomimetic complexes. So for aerobic organism, you know that O2 plays an important role. I mean, it's actually playing a pivotal role in carrying out many oxygen-dependent transformation reactions and metabolically relevant transformation reactions. So if we just go to the basics of this very simple but very complicated molecule Mother Nature gifted us, is the O2. And if we just look at the reaction of any organic compound with oxygen, at the end, eventually after several steps, it forms carbon dioxide that we breathe out. You have water, and for every step or every molecule of this glucose where it requires 6 equivalent of oxygen, it produces about 3000 kilojoule per mole of energy, and which is actually a huge amount of energy. And that's why aerobic organism survives over anaerobic species in the process of evolution. But this reaction is thermodynamically favorable. As you can see, it's actually producing a large amount of energy. But this reaction is not kinetically favorable. The reason is very simple. As all of you know that we can see this, its manifestation is obvious, that it is not kinetically very reactive, it's kinetically sluggish, and that's why organic substances around us, they coexist with oxygen instead of getting burnt to carbon dioxide and water. And this particular lack of reactivity in terms of the kinetic sluggishness for this reaction is due to its interesting electronic structure. And let me take you to our basic first year undergraduate chemistry, the molecular orbital of O2, the dioxygen molecular orbital. So if we just look at the molecular orbital, and if we just focus on this part here, this is the antibonding molecular orbitals. So in the ground state, the antibonding molecular orbitals, both the pi star antibonding orbitals are singly occupied. And this is according to Hund's rule of maximum multiplicity. This is the ground state. But definitely there is an excited state where the Hund's rule of maximum multiplicity does not apply. So if I just look into that one, the spin state of oxygen can be represented as triplet, because I have 2 unpaired electron, and that gives your S is total S equal to 1, and therefore its multiplicity will be twice S plus 1, so that would be triplet. Now if we look into the organic molecules, where most of the organic molecules, unless they are radical species, they are all singlet, because there is no unpaired electron present there. They are all paired up electron in the form of covalent bond. So this singlet organic molecule react with a triplet oxygen molecule to produce carbon dioxide and water. Again, if we look to the structure of carbon dioxide, the electronic structure of carbon dioxide and of water, in each case, there is no unpaired electron. So therefore, both of them are singlet. So in this reaction, if we just look at it, a singlet organic molecule is reacting with a triplet oxygen. Overall, it is forming carbon dioxide, which is singlet, and water, which is singlet. So in this reaction, what is not conserved? It is the spin multiplicity is not conserved in this reaction. And this particular spin issue, the spin multiplicity, which is not conserved, and this restriction actually restricts its reactivity by imposing a kinetic barrier, what I said. And therefore, oxygen in its ground state, which is triplet, is not reactive. It has to be activated to make it reactive. So in living system, in biological systems, oxygen is activated by different pathways. But what are the other methods of oxygen activation? If we understand that part, then I think we'll understand why Mother Nature has opted a particular path which involves different transition metal ions to activate oxygen from its relatively less reactive triplet state to more reactive singlet or other reactive state. So if we just look into the different pathways by which oxygen can be activated, as you can see, the triplet oxygen can be photochemically activated to its reactive singlet state, or I can make oxygen reactive by removing an electron to form a cation, or I can dissociate oxygen to form, 2 oxygen atoms. But if we look at the biological systems, does not like this photochemical oxidation, photochemical activation process, or either oxidative or dissociation pathway, because both these pathways involves very high energy pathway or very high energy reaction system. Whereas oxygen can be activated through reduction process, what is called reductive activation. So if we just think of that, if I give one electron here, if I inject one electron, the electron will go to anti-bonding molecular orbitals. And precisely that gives you a bond order of 1.5, and that's nothing but called a superoxide species. Now, if I inject a second electron, that second electron will also go to the anti-bonding molecular orbital, and now your bond order is 1. So instead of 2, now your bond order is 1, which is nothing but a peroxide. Now I still have 2 electrons that I can inject to O2, and those 2 electrons can go to the sigma star molecular orbitals of oxygen. So overall, I can reduce oxygen by 4 electrons from oxygen to generate water. So in the first step, you have superoxide. The second electron gives you peroxide, and then further 2 electrons will form water, or you can write it in the form of oxides where OO bond no longer exists. So in the ground state triplet to the excited state singlet, what you call, they are of in a high energy difference of about 100 kilojoule. And therefore, biological systems normally takes care of the dioxygen activation through reductive activation pathway, because photochemically activation requires photoactivator and light. Ionization requires very high energy, as you can see here, because removing one electron from oxygen, it's a very energy demanding process, and therefore ionization energy will be very high, and its ionization energy is almost equal to the ionization energy of a xenon atom. So therefore it's not at all energetically feasible, because after all, at the end of the day, the oxygen is required in biological system to produce energy, not to use that energy for the activation of oxygen. And as well, the dissociation of the oxygen molecule is also very highly energetic process, and it requires burning at very high temperature. So both these pathways we can discard. So what happens in biological systems? In biological systems, in the process of evolution, different metal ions, depending upon their abundances, depending upon their availability and bioavailability, different metal ions were picked up by enzymes. And those enzymes are called metalloenzymes. That means it's a metal containing enzymes that perform many catalytic reactions in living organisms. So those are nothing but those metal complexes can form metal ligand complex, where in biological system you have ligand like porphyrines, you have amino acid side chains as ligands, and they can bind oxygen either reversibly or irreversibly. So overall, I can write down that the biocoordination compound, that means coordination compound with proteins or other bioactive ligands, they can form this MLN kind of metal complex, which can react with oxygen, and they can bind oxygen reversibly, just like hemoglobin in blood, which involves this particular step, which is a reversible process. Now, if I consider that the reversibility, the equilibrium constant, if I consider this as the capital K, there could be another process if whether this would get back there or it would react further. That depends on what is the rate of the second reaction when it reacts further instead of going back to the metal ligand complex, which exactly takes place in hemoglobin. So this capital K and small k dictates whether it would be an oxygen binding and transport protein, or they would perform some kind of oxygenation reactions in living system to catalyze the many important metabolically relevant reactions. So these two extreme possibilities include where K is very high and the small k is low, then they are oxygen carrier complexes like hemoglobin, myoglobin, hemocyanin. If K is small, but this small k is high, then basically it's more like a redox type of reactions where oxygen undergoes several steps of reduction and then eventually it forms reactive oxygen species which is particularly implicated or which are implicated as the active oxidizing species in biological system. So those enzymes include oxygenases or oxidases, which I will come in a moment. What are they? So in general, in biological systems, the dioxygen activations involves different transition metal ions because transition metal ions have unpaired electron. And therefore, these unpaired electrons of transition metal can control the electronic configuration of oxygen. So they can pair up, the transition metal unpaired electron can be paired up in the form of metal oxygen complex, and they can form metal oxygen complex where you can see that you have one unpaired electron up, another unpaired electron down, and eventually this gives you S equal to 0 when oxygen binds to the metal center, and therefore now the oxygen is activated on the metal center. And this oxygen activation process, many different intermediates are formed, as I already mentioned, that in the process of equilibrium, that means a reversible binding of oxygen, oxygen can bind just simple metal oxygen adduct. It can form metal superoxide species where metal gives up electron to oxygen, metal center gets one electron oxidized, oxygen gets one electron reduced to superoxide, or you can have your metal center providing 2 electron to oxygen where metal center gets 2 electron oxidized and oxygen gets 2 electrons reduced to peroxide species, or you can have 2 metal centers each contributing 1 electron to oxygen. That means oxygen will get 2 electron reduced, but each metal center will get 1 electron oxidant, and therefore it forms a peroxo-based dimeric species like this. So these are the species that form initially depending upon the enzyme, depending upon the reaction, or depending upon the choice by Mother Nature for what this particular enzyme has been developed in living organism. So these reactions in biology, as I said, that when the reaction does not involve the reversible binding, but if your small k, as I have shown in the previous slide, your small k is higher than the capital K, then several reaction takes place. Oxygen is reduced either to peroxide or water, depending on the actual oxidation reactions or the purpose for which this enzyme is involved. And in this process, you see the substrate is getting oxidized and oxygen is getting reduced either by 2 electron to hydrogen peroxide or by 4 electron to water. But in the process, substrate is getting oxidized. So the electron is actually removed from the sulfur, from the substrate to oxygen, substrate gets oxidized. But in the process, no oxygen atom from O2 is incorporated into the substrate. And these type of reactions are called oxidases or oxidation reactions, and the enzymes that perform these kind of reactions are called oxidases. Whereas there is another kind of enzymes that not only perform oxidation reaction, but this oxidation reaction involves the incorporation of 1 or 2 oxygen atom from O2, and these are called oxygenases. So that means oxygen atoms are incorporated into the product in the process of oxidation. So this oxygen atom is derived from O2, and this, since 1 oxygen atom is coming from O2 molecule, this is called monooxygenase. Whereas if both the oxygen atoms of the product comes from O2, then this is called dioxygenase. And nature primarily involves these oxidases or oxygenases depending on the purpose, depending on the reaction they catalyze. Now, what I said, and I promised that I would take you through the kind of role of synthetic metal compounds and how they perform these reactions, and from these synthetic compounds, what information we can get about the mechanism of enzymatic reactions, that how enzymes perform these reactions, what are the intermediates, what are the actual oxidizing agent, because as I have already pointed out that O2 cannot be the oxidant. O2 is the terminal oxidant, but in the process, O2 is reductively activated and then it forms many different intermediates. The most reactive species will be the oxidizing agent, and that perform the substrate oxidation or oxygenation, not the O2. So what are they? Can we get any information from synthetic system? So before going to that, let us see, since my group is primarily interested in understanding the reactions that are catalyzed by iron-containing enzymes. And iron enzymes are ubiquitous, particularly in higher organisms. And you can see that they perform a myriad of transformation reactions, starting from oxidation, oxygenation reactions that include epoxidation, CH bond hydroxylation reactions, cis dihydroxylation reaction, ring formation reaction, aromatic or aliphatic CC bond cleavage reactions, and electron transfer reactions. So as you can see that this includes almost all the reactions that we normally deal in synthetic chemistry, but in biological system, these reactions take place under ambient condition using oxygen from air as the sole terminal oxidant, and iron containing enzymes are the catalyst. And it is the ultimate green process by which Mother Nature has optimized these reactions in the process of billions of years of evolution process. And these metal containing proteins, basically they not only control the O2 molecule for its activation from triplet state to its more reactive state, but in addition to that, it also performs several other reactions just to avoid the formation of any side product, because as I have told that sometime, because nothing is perfect, so there is some sort of imperfection in living system also, which is nothing but manifestation in form of disease. And this reductive activation process, sometimes it forms reactive oxygen species like hydroxyl radical hydrogen peroxide, or sometimes free superoxide not coordinated to the metal center, and they are freely diffusing radical. If you have a freely diffusing radical, they can perform uncontrolled oxidation and therefore cleaving the DNA, cell, and many other organic reactions that they can perform in living system. But nature has a very well synchronized strategy that they can also deal with this kind of deleterious side reactions and have control on these reactions. So other than normal oxidation, oxygenation, ring formation, CC bond cleavage, biosynthesis of different metabolites, biosynthesis of antibiotics, or many other process, it also takes care of the side effect of oxygen, which also just cannot be ruled out. When that process dominates, we actually see that in form of disease. So let us show you some representative examples, because I just cannot, because the list is endless. So I'll show you particularly, which are relevant to today's discussion, some representative example of oxygen activating iron enzymes. You are all familiar with heme containing proteins like hemoglobin, where your iron center resides in the macrocyclic cavity of a porphyrin ligand. And this enzyme, particularly in case of hemoglobin, which is a protein, not an enzyme. Its role is just oxygen as an oxygen carrier. But in case of oxygenation or oxidation reactions, porphyrin, iron, metalloproteins are also involved, and they are enzymes, and they are in the group of some examples like cytochrome P450 group of enzymes. What they do, they perform, you see, a CH bond hydroxylation reaction of very, very refractive CH bonds. Very strong CH bonds can be oxygenated. And in the process, oxygen atom, one oxygen atom from O2 is incorporated into the hydroxylated product, and another oxygen atom from oxygen lands up as water. And here you see, for the process, you need electron, because that's why it's called reductive activation. And these two electrons are provided by electron transfer protein like cytochrome C oxidase or some other. This group of superfamily of enzymes, they also perform the epoxidation of alkenes, and they are very crucial not only in the metabolism of drug, but also metabolism in many other functions in living system. So these type of proteins are called heme proteins, because you have a heme iron porphyrin unit in the active site of the protein. Whereas there are another group of enzymes completely different from heme. These are called non-heme, because they are not heme. And here you see that a metal center can be coordinated by the side chains of amino acid residues, and they bind to the metal center. And this particular site is called the active site of the protein, or in this case, it's enzyme. And here actual chemistry takes place. The protein structure provides a kind of a channel, or the conformation of the protein allows the reaction to take place in a synthetically more chemo, regio, and stereoselective manner. So this kind of non-heme iron enzymes family, some examples like alpha keto acid dependent oxygenases. What is this? As I told that here in the heme system, you need electron, and these electrons are provided by some electron transfer proteins. In case of non-heme oxygenases, there are some enzymes that require this electron also for the activation of triplet oxygen. But here the electrons are provided by organic cofactor, and that's why they are called alpha keto. So this is an alpha keto acid, and these alpha keto acid dependent oxygenases, they involve the oxidation of alpha keto acid to carboxylic acid, and the electron which are ejected in the process are actually used for the activation of oxygen on the metal center, on the iron center. And then the reactive iron oxygen species, the actual active iron oxygen species, is involved for the oxygenation of CH bond, leading to the generation of ROH. And here this oxygen atom is derived from oxygen here, from O2 molecule. And interestingly, you see that another oxygen atom is incorporated into the carboxylic acid, which actually originates from the alpha keto acid with concomitant elimination of carbon dioxide. So this is a process which is called oxidative decarboxylation of alpha keto acid, and the electrons are utilized by the oxygen for its reduction, which performs the aliphatic CH bond hydroxylation reaction. Another very interesting oxygenases I must tell you, and our talk today involves a lot on this particular one, is called risky dioxygenases. And these risky dioxygenases are the enzymes that are found particularly in soil bacteria, and their role is just to cleave the toxic aromatic compounds. And in these toxic aromatic compounds, in the first step, what they do oxygenation on the relatively less reactive aromatic substrate. So if this is, you can look at this one, you have a double bond, and moreover, this is a very stable aromatic system. But nature has created this enzyme for a purpose that these toxic compounds need to be degraded. So the first step which is required is to do some functionalization on this relatively inert substrate. And that these enzymes perform the cis dihydroxylation on the double bond and perform and shows this, you see here, a cis dihydroxylated product. Now if I go and talk about a little bit of basics organic chemistry that we normally teach to our undergraduate students, that cis dihydroxylation reactions involve, like, it requires a very toxic reagents like osmium tetroxide and some other related reagent, but they need it in stoichiometric amount. So in the reaction, the side products that you get are actually very toxic for the environment. But here you see that in the reaction, you are forming a cis dihydroxylated product, then it can further undergo aromatization. And once this again forms an aromatization, this is an 1,2 diol product on the aromatic ring, most like a catechol. And then this catechol can be cleaved very easily, which can be utilized by the soil bacteria to form, right, to form. Just a second. Just a second. No, talk. I'm giving a lecture here, okay? All right. So sorry for the disruption. So this particular compound, you see, this reaction is a unique reaction because this reaction takes place under ambient condition, and it is performing cis dihydroxylation on the double bond, and that too of the aromatic ring. And this is completely an atomic process. There is no waste. Both the oxygen atoms from O2 are incorporated here. And in this process, you need 2 electrons for the reduction of oxygen. And these 2 electrons are provided by some iron sulfur proteins, which are called risky proteins. And that's why this group of oxygen enzymes are called risky dioxygenases. Now, how these enzymes perform these reactions? As I said already, that neither O2 nor superoxide, not peroxide are the active oxidizing agent. So what is the active oxidant? O2 is the terminal oxidant. I repeat, oxygen is not the active oxidizing agent. So how this triplet oxygen is activated and leading to the generation of active species to carry out such intriguing reactions that are really challenging for synthetic chemists. And can we emulate these reactions in synthetic system? For that, if we understand the mechanism of these reactions, I think that would help us to design and develop and synthesize such kind of synthetic catalyst in in synthetic laboratory. Now, most like synthetic studies, spectroscopic studies, enzyme studies, the accumulated information that are available to us provides an information that iron 2, which is present in the active site, in most of these cases, iron is in +2 oxidation state. So if it is in a +2 oxidation state, this is the reduced form of iron. And this is called the resting state of the enzyme, where enzyme takes rest. And then this iron center then reacts with oxygen, where iron center provides electron to the antibonding molecular orbital of O2, initially forms this species, an iron 3 superoxide species. Then this superoxide species, which is actually a common intermediate, depending on the nature of the reaction, depending on the enzyme, it actually diverges to different pathway. And in one of the pathways where there is an organic cofactor, the one I have shown you, the alpha ketoglutarate or alpha keto acid dependent oxygenases, where cofactor provides the electron to the metal center, or an electron from electron transfer proteins in the presence of proton it can form. So if I add one more electron, that means here it will form peroxide. In the presence of proton or a cofactor, this can form an iron 3 hydroperoxo or alkyl peroxo species. And this iron 3 hydroperoxo species can undergo OO bond cleavage. And this OO bond cleavage can take place by two different pathways, depending on several parameters. The spin state of iron center plays an important role. And here in this case, if it goes on OO bond hydrolysis pathway, that means your iron center provides 2 electrons here, and this bond is hydrolytically cleaved. So both the electrons will go to OH, and therefore it forms either OH minus or OR minus, and iron center is providing 2 electrons, so it can form an iron 5 oxo species. And this iron 5 oxo species are the active oxidizing agent that perform the oxidation reactions. So this is the actual oxidant or oxidizing agent. It may so happen that this OO bond, depending on the spin state of iron center, can undergo homolytic cleavage as well. So if it undergoes homolytic cleavage, that means one electron comes here, another electron goes there, and in the process, this comes out as an hydroxyl radical. But iron center then provides 1 electron, and it then forms iron 3 is converted to iron 4. So you now have an iron 4 oxo species, which is the active oxidant. If this species gets 1 electron reduced, and iron center then come back to iron 2, then OO bond can also be cleaved hydrolytically. Then it forms again, hydrolytic cleavage will form an iron 4 oxo species. And another pathway which takes place quite often when a heme system does not perform reversible oxygen binding, but irreversible oxygen binding, when the protein, the heme protein size becomes smaller, it can undergo a dimeric pathway. And then you can have this peroxo-bridge dimer, as I mentioned before, that another iron center can provide the second electron for the reduction of oxygen to peroxide. It can form a peroxo-bridged diiron 3 compound, and then this OO bond can be homolytically cleaved, leading to the generation of 2 equivalent of iron 4 oxo species. And trust me, and as I will show you while I progress, that these are the active oxidant, and they indeed are possible. So don't get confused that iron 4, iron 5, are they possible? Why not? If iron 6 is possible, like ferrate, which is a mineral that is found in a water-free environment in deep mines, where iron is in +6 oxidation state. And it's a very reactive species. Many of the undergraduate students, all of you know, who have studied already the Frost diagram or predominance diagram in your redox chemistry lecture. There you have seen that iron 6 could be very, very reactive species, and you cannot just keep it in water. It will oxidize water immediately because of its very high reduction potential of the iron 6 to iron 3. Okay, so now let us look what are the active oxidant in these enzymes. So the information that is now available, where it says that in this case, alpha keto acid dependent oxygenases, an iron 4 oxo species is the active oxidant. And this has now been spectroscopically characterized. A few milliseconds is the lifetime of this species, and therefore it's an intermediate, very reactive species. More reactive a species means it is less likely to be absorbed. But I think as a researcher, it is our objective to see those intermediates and try to understand their electronic structure, their reactivity, their properties, and so on and so forth, so that it helps us to design very good and reactive catalyst. In case of risky dioxygenases, the intermediate which has been characterized long back in 2003, you can see here, which is an side-on bound peroxo species. So it's an iron 3 peroxo species which has been crystallographically characterized, but there is still debate whether this is the active oxidizing species that is involved in the cis dihydroxylation reaction, or high valent species like iron 4 or iron 5 species, oxo species, are the active oxidant. And in case of heme protein like cytochrome P450 superfamily of enzymes, it's well established that an iron 4 oxo intermediate, or sorry, it's an iron 5 oxo intermediate, or formally you can say it's an iron 4 oxo and porphyrin radical cation, is the active oxidant in this group of oxygenases. So how Mother Nature controls this kind of reactions and perform stereo, regio, and chemoselective reactions, how the metalloprotein active sites bind oxygen, and how this activation process takes place at really some rate which is otherwise not possible to achieve with synthetic system. And as I have said, and I always appreciate the kind of beauty in the design aspect in the process of evolution, where such kind of reactions are performed with really an exquisite control on the reactivity. So how do we know this? As I said, enzymatic studies provide useful insights into the nature of the reactivity by these enzymes. But quite often, enzymatic studies are difficult to perform because purification, isolation of proteins are quite difficult and handling them quite difficult. And moreover, they are sometimes, do not give you quite good information, because if you have a reactive species with enzymatic system, you need to deal with aqueous solution, and quite often they tend to degrade. So therefore, working with the enzymatic system is not always easy. And moreover, you have to have expertise. So now, as a synthetic inorganic chemist, I, with formal training in organic chemistry, what we can do? We can actually develop synthetic systems which are very similar to the enzymatic system, except the protein part, that only focus on the active site part. And then we can develop synthetic compounds, which are called model compounds. And then we'll try to understand whether these model compounds can provide some information about the enzymatic reactions, their mechanism, which would eventually help us to develop catalytic systems, not exactly as that of the enzymes, but close to the enzymatic systems. So the approach towards that is like synthetically how we can model those. So in bioinorganic chemistry, where the metal center, as I said, is the active site is coordinated by amino acid side chains, and they provide a particular geometry. And this geometry is very, very important because the reduction potential at the active site of the metal depends on the geometry. And therefore, what we do, we look at the active site geometry of the metalloenzymes. We do not work with metalloenzymes. We work to understand that what is the geometry at the active site, what are the different groups, amino acid side chains that are coordinated to the iron center, and accordingly, we try to develop synthetic compounds. And here you can see, we may consider from a coordination chemistry point of view that these amino acid side chains are nothing but these are ligands. So we have to design ligand, and then we have to prepare the synthetic compounds. And these are called synthetic models. And these synthetic models in biomimetic chemistry to mimic the enzymatic reactions of two types. They could be structural. That means I can get a model compound which is very close to the structure of the active site, but it's not reactive. But in fact, if we do oxidation, if we perform structural studies, spectroscopic studies, we can get some information about the oxidation state, electronic structure, coordination environment, and also the stereochemistry. But primarily, my research group is interested in functional modeling. That means we try to emulate the reactions using the synthetic systems and to emulate the reaction which is taking place in enzymatic system, like the CH bond hydroxylation reaction or sister hydroxylation reaction. And for that purpose, we just prepare coordination compounds, the model compounds, and we'll see first that what is the active site geometry of the synthetic models. And here you see in the iron center, which is in the resting state, is 6 coordinate. So it's a 6 coordinate iron center, and in one face of the pseudo-octahedral geometry of the iron center, 3 ligands are there. So it is actually the ligands are coordinated in facial manner, and other labile ligands, because this water can easily be replaced by substrate or by oxygen, where actual chemistry takes place. So for that purpose, we need to first design a facial ligand that can bind to the metal center from one face. And this is just a cartoon which shows that this kind of ligands would be very useful to mimic the active site structure with a facial coordination motif. So in that direction, our strategy is to use very different kind of ligands, like prepare ternary compound. And obviously our inspiration comes from biological system, where in the alpha keto acid dependent oxygenases, the enzyme and cofactor adapt, where cofactor also act as a co-ligand. And then the reaction takes place because this is a 5 coordinate iron, here this is water is there, which is labile. So water can easily be replaced. Oxygen can go there to the iron center, and then the reaction starts. So taking inspiration from this kind of an enzyme cofactor adapt, we also utilize a similar strategy of using ternary metal complexes. Ternary metal complexes means you will have a ligand which will support the metal compound. So you have a metal ligand complex, and then you need a reductant which will provide the electron for the reduction of oxygen. So this reductant is, we call it a sacrificial reductant. That means after one cycle, this L prime will get oxidized, but in the process, the electron which comes out from L prime will go to oxygen, and oxygen will get activated in the reductive activation pathway. And for that, if you do that, then this metal ligand complex can selectively transfer electron, because the problem is what happens if you take L prime outside, the L prime, depending on the redox potential matching, the L prime can provide electron either to the metal center, or it can provide electron to the oxygen, or it can provide electron to the free oxygen which is in solution, in an oxygen saturated solution. So that means it may give rise to an uncontrolled electron transfer process. So to avoid the uncontrolled electron transfer process, the use of ternary metal complex was anticipated to provide a more selective electron transfer pathway. And let us see what exactly happens in this system. So we prepare this kind of iron complexes, where we prepare ternary iron compound, like we have a supporting ligand, a facial, where 1 nitrogen, 2 nitrogen, and 3 nitrogen, they can bind to the iron center. And today I'll show you that the sacrificial reductant, like alpha hydroxy acid, which are 2 electron sacrificial reductant, because this can undergo decarboxylation, and this electron will be utilized for the reduction of oxygen on the iron 2 center. So in that direction, we have prepared the ternary iron 2 compound, the first compound long back when I started my independent career back in 2007. But it took about 2 to 3 years to isolate this compound, and I had really a very bright PhD student at that time, who is now a faculty at IIT Delhi, who in fact took a lot of pain to isolate the cis crystals, because these compounds are highly air sensitive, because we need to prepare compounds that are air sensitive. We cannot prepare anything outside. So we need an inert atmosphere system where we can do this synthesis. And here you see this is an iron center coordinated by the nitrogen donor ligand of this 3-spirazolyl borate ligand. And then you have an alpha hydroxy acid, which is basically a mandelic acid, which is here coordinated through the carboxylate oxygen. This is actually a high spin iron 2 compound, as you can see here from the optical spectral feature, which is almost featureless, typical of the high spin iron 2 center. But this compound is in benzene. If I dissolve this compound in benzene and expose to O2, what it does, the colorless solution slowly turns green. And it actually a few seconds that it requires to turn to green. And the green solution shows an EPR spectrum, which is typical of a rhombic signal, typical of a high spin iron 3 compound. Well, iron 2 compound I start from, I go to iron 3. Quite often we tend to discard this because this is nothing but a wastage of time. But then what we did, we tried to thought of that what is happening, let us investigate into that. We took the reaction solution and then we analyze the reaction solution by ESI mass spectrometry. And the mass spectrum suggests that I have a peak here at mass of 740. And this 740 peak then, in fact, matches with an iron compound of this one, but that we did not start with. This hydroxide group comes on the one of the aromatic ring. So what happens in the process? There is an intramolecular ligand hydroxylation that took place, and the green chromophore is actually coming. Since it's an iron 3, the green chromophore is coming from the phenolate to iron 3 charge transfer transition. So I have a phenolate iron 3 charge transfer transition. What happens? In addition to that peak, I have another peak, which is at 884. And this corresponds to the hydroxylated ligand, iron, and a benzoate. So where the benzoate come from? Because as I said, the alpha hydroxy acid can undergo decarboxylation. And we can see here the formation of different products like benzaldehyde, benzyl alcohol, and benzoic acid, all sorts of products that we have here. Here your major product is aldehyde. Can you please let me know when the time is up? So if you alert me, then it would be good. Otherwise, I have a bad habit of continuing things for, without looking at the watch. So
it is almost time.
It's almost done. So I can stop at any moment, but I had a lot of introductory things for the sake of students. So just now I am slowly entering into research, maybe in another 10 minutes.
Yes, sir. Yes, sir. All right.
If it is not a problem.
No. Please continue.
Okay, fine. So here you see that we get like aldehyde, we get alcohol, benzyl alcohol, and benzoic acid. This was pretty puzzling at that point. What we did then, we then performed the same reaction with isotope leveled oxygen. So instead of normal O2, we used 18 leveled oxygen. So 18 isotope leveled oxygen, we now see that 740 peak that we observed with normal oxygen, that means 16 O2, is now shifted from 740 to 742. That means this product, the iron 3 phenolate product, this oxygen on the phenol ring originates from O2. And the 884 peak now shifted to 888. That means not only this phenolate product in this hydroxylated ligand, but also the benzoate, which is derived from the alpha hydroxy acid mandolate, also contains one of the oxygen atom. And that was the beginning of the whole chemistry that subsequently we performed for the last several years, which in fact yielded quite a useful insight, provided useful insights into the reaction mechanism. Then what we did that not only the benzyl alcohol, benzoic acid, but benzyl alcohol show peak in the presence of O18, not only for at 108 for the benzyl alcohol, but benzyl alcohol oxygen also originates from O2. So some partial incorporation of oxygen atom into benzyl alcohol was also observed. So in a nutshell, what was observed that one of the oxygen atoms of benzoate contains 1 leveled oxygen. a small percentage of level oxygen is incorporated into benzyl alcohol, because here it is not only 108 peak, but you have a 110. Had it been 100% O18 incorporated, then I would see only 110. That means this is a partial incorporation of oxygen into benzyl alcohol, but benzaldehyde does not contain any leveled oxygen. And this was at that point quite puzzling. So we tried to simplify the system, and then we again went back there, that instead of mandelic acid, now we have used a benzylic acid, where it is still an alpha hydroxy acid, but instead of an alpha hydrogen here, now I have a phenyl group. So we did the same reaction with this system. This is the crystal structure of the 5-coordinate high-spin iron 2 benzylate compound. And in this case, we also perform the reaction with oxygen like this 740 peak, and then we shifts in the 2 mass unit to 742 in the presence of isotope level oxygen. And this particular unit, the alpha hydroxy acid unit benzylate, is oxidatively decarboxylated to benzophenone quantitatively. So unlike the previous case, which was relatively complicated, but when you have a complication means there is a beautiful chemistry there. So we should investigate that. But this particular finding helped us to understand that, oh yeah, so in case of a mandolate, if I have an hydrogen here, it is expected that this should give you a benzaldehyde as the only product. But I don't have only benzaldehyde, I have benzoic acid and benzyl alcohol. So what is happening there? Then we try to perform a lot of mechanistic studies using radical quencher, and we found that if I have an iron 3 superoxo, which is formed initially, if it abstracts an hydrogen atom from the OH group, because this group is very, very important. If instead of alpha hydroxy acid, if I take alpha methoxy species, this does not react with oxygen at all, they are rock. So this alpha hydrogen atom is probably involved. But can we do one thing? Instead of doing this reaction and abstracts the hydrogen atom by the superoxide from this group, if I add an weak OH substrate in excess amount, then there will be a competition between intramolecular hydrogen atom abstraction versus intermolecular hydrogen atom abstraction, and that too from a weak OH bond. So we used temp OH, and we found that this decarboxylation of benzylate is inhibited. So therefore, OH abstraction by the initially formed iron 3 superoxo species is probably initiate the reaction. And as I said, if I use instead of alpha hydroxy acid, if we used alpha methoxy species, without or with alpha hydrogen, they do not react with oxygen. Like this compound, you can keep it for years, although it's an iron 2 compound under oxygen. So therefore, the presence of an OH group is very, very important for the reductive activation of dioxygen and subsequent reaction. Then we tried to find out what is the active oxidant, what is there, who is performing the aromatic ring hydroxylation here. So for that, we did again this different substrate oxidation, intramolecular, in contrast to the intramolecular ligand hydroxylation or phenyl ring hydroxylation, I did intermolecular substrate oxidation. And here you see that when we perform the reaction with thioanisole, like phenyl S-methyl, and we see that in the absence of any substrate, I have complete ligand hydroxylation, because we can easily quantify that from the chromophore, which is here in the 600 nanometer region. But as soon as I add thioanisole, depending on the amount of thioanisole here, the ligand hydroxylation gets inhibited. That means my substrate thioanisole is getting oxidized. And indeed, it forms a mixture of sulfoxide and sulfone. In the presence of O18, it forms an oxygen incorporated peak. Instead of 140, I have 142 peak, 156 peak is shifted to 160. Perfectly all right, that these are all originate from O2 molecule. Then we did some kind of a Hammett analysis, and that provides us an information that the active oxidant, which is responsible for thioanisole oxidation, is nucleophilic in nature, because this Hammett sigma versus the relative rate, logarithm of the relative rate for the substrate oxidation gives you a positive slope with a different substituent on the thioanisole from electron withdrawing to electron donating groups, and it's a linear correlation with a positive slope. That means it is actually the substrate involved is nucleophilic in character. Can it perform some other chemistry? Yes. Not only that it performs oxygen atom transfer to thionisole, the most intriguing observation that Sayantan came up with back in 2010, that when he added an alkene, say for example, cyclohexene, or 1-octene, or cyclooctene, or styrene, what he found is that selectively he got 1,2-diol product, the cis-diol product as the only product from alkene. And this was really pretty exciting. And here you see that when the reaction, this particular cyclohexane 1,2-diol shows a peak at 116. Now the same reaction in the presence of isotope leveled oxygen, 18 isotope leveled oxygen, the 116 peak is shifted. It's a small peak here you can see at 120. Neat. So if you just zoom it, you can see that this is an 120 peak, which is the molecular ion peak, which is nothing but both the oxygen atoms of the diol are actually originating from oxygen. So just like risky dioxygenase chemistry, we can actually see the cis hydroxylation of alkene using oxygen from air. And therefore, that was precisely allowed us to venture into a completely new area of how we can use oxygen for doing catalytic oxidation chemistry, either cis hydroxylation chemistry or other chemistry. So as I said, that we predict that this should be a nucleophilic oxidant which is performing the cis hydroxylation reaction. But then what happens? Let us understand, we go back again to the previous. Now, if I have the oxidant and if I add benzaldehyde to it, what happens? Instead, so interestingly, we took the iron to benzylate complex and now we added benzaldehyde or substituted benzaldehyde as substrate. And here we got a mixture of benzyl alcohol and benzoic acid. That was precisely observed with mandelic acid. So first, definitely, as expected, benzaldehyde was forming, but then the benzaldehyde was transforming to a mixture of benzaldehyde and benzoic acid and benzyl alcohol. An isotope leveling experiment perfectly matches with this thing. And then we did some kind of a Mandelate D kind of mechanistic studies with, instead of alpha hydrogen here, we took a deuterated Mandelate and then reacted with oxygen. What we found is that the same thing, that benzyl alcohol or get 2D from this D. And benzoic acid does not contain any D because there is no way of having any D because this D is in the benzylic position. And this tells us something. If I start from a benzaldehyde and if I get benzyl alcohol and benzoic acid, this tells us that this is a chemistry that we all study in our undergraduate studies, which is called Cannizzaro reaction. And Cannizzaro reaction takes place in the presence of an hydroxide ion. So what happens, I'll just stop with this slide. I can continue, but I will stop with this slide with at least one story that I could tell you, that this hydroxide is coming from where? So all our mechanistic studies point and indicate that initially my iron 2 compound reacts with oxygen, and in the process, the alpha hydroxy acid undergoes decarboxylation with concomitant formation of a ketone. If R is H, it can form aldehyde, which I'll come in a moment. And in the process, since it's a 2-electron reductant, oxygen is 2 electrons reduced to form this peroxo species. This peroxo species then can undergo, if this one is a nucleophilic in nature, and benzaldehyde, when I have this R equal to H, this is nothing but benzaldehyde. So now this benzaldehyde goes there, acts as a substrate. So a product in the first step acts as a substrate, and this center is an electrophilic center. So you have a nucleophile, a nucleophilic iron peroxide intermediate. It will attack on this one, and then it can form a benzoic acid with a metal coordinated hydroxide. This metal coordinated hydroxide is a strong nucleophile again as a base. So this hydroxide then can attack the benzaldehyde, another equivalent of benzaldehyde, and then it actually forms this gem diol product. then this gem diol, which can remain in equilibria because this can eliminate hydroxide, and therefore it can explain why you have a partially leveled oxygen into benzyl alcohol. Because this hydride migration in the Cannizzaro reaction, it's already this benzaldehyde is partially leveled. And this hydride, when it attacks at this center, the migration, it gives you benzyl alcohol will partially leveled, not fully leveled. And similarly, benzoic acid also give you a partially leveled oxygenated product here. So it's basically a Cannizzaro type reaction that is taking place, but actually it undergoes oxidative decarboxylation to form benzaldehyde in case of mandolate. In case of benzylate, it forms benzophenone. But in both the cases, you have an iron 2 hydroperoxide. In case of mandolate, the first product is actually a substrate, and therefore substrate oxidation cannot be carried out. In case of benzylate, since there is no substrate, because benzophenone cannot act as a substrate, so in the presence of external substrate, that external substrate performs the reaction, and that gives you either cis hydroxylated product or oxygen atom transfer product. So what I will just end up the story by showing you here that what we found is that an iron 2 alpha hydroxy acid, the ternary compound, activate oxygen to undergo decarboxylation of the metal coordinated co-reductant or sacrificial reductant. Then it can form the active species, which is iron 2 hydroperoxo species, which can perform cis hydroxylation reaction, or it can do sulfur oxidation reaction. What I could not show you, that in the presence of Lewis acid, I can convert this nucleophilic oxidant to an electrophilic oxidant, which can generate an iron 4 oxohydroxo species. This can also perform cis dihydroxylation reaction, but this particular species can exchange its oxygen atom with water. And other than the cis dihydroxylation reaction, this can also perform aliphatic CH bond hydroxylation reaction. And this CH bond hydroxylation reaction chemistry and all this chemistry in the presence of Lewis acid and protic acid, we have recently, wrote it in a special book chapter, which is will come up as a comprehensive coordination chemistry series that is reflected that where what exactly kind of insight that we could provide in this particular reaction. But these intermediates, since they are highly reactive, we could not trap them. But very recently, we have, in fact have the results that we have. I don't want to get into those. We in fact have recently shown that using Lewis acid, we can in fact trap those oxidants and we can make them less reactive, and then we can see them at low temperature, and then we get information about their electronic structure and other properties. So in a nutshell, what I wanted to tell you that our inspiration was from biology, but by using the common sense of synthetic chemistry and organic chemistry, spectroscopy helped us to understand the mechanism that is taking place in biological systems. And moreover, this would open up new avenues for the synthesis of a catalyst, which are metal-based catalysts for performing CH bond hydroxylation reactions or carbon cis bond cis hydroxylation reaction using oxygen from air, which is cheap. You don't need to pay anything for that. And eventually at the end, you do not have any toxic side products. So this is what I wanted to share with you. Whatever I told you or presented you, particularly the research part, not the first part, it was very easy for me to present, but it involves a lot of hard work by my co-workers with a very hard-working, intelligent group of people who work with me and are working. So this work, the first part was performed by Shayanton, who was in fact the first group of PhD student in my group, who is now an assistant professor at IIT Delhi. And then this work was contributed to this work. Contributions also came from Vishwarup and Shayanti. Vishwarup is also in IIT Delhi as a faculty. Shayanti is in Germany now. She has been she was instrumental in understanding the mechanism for the electrophilic oxidant part. The others also contributed some way or other. These are the group members, my collaborators, particularly Professor Ankon Pal, my colleague here, and his former PhD student Gaurav helped us a lot from theoretical studies. The most important part is funding, because this kind of research requires a lot of money. And I'm fortunate to have such kind of funding from many sources like Department of Science and Technology, Government of India, through Science and Engineering Research Board, Indian National Science Academy. They give us funding for in Indian National Science Academy Young Scientist Award project, which I received about 10 years ago. And then Council of Scientific and Industrial Research, and I had a foreign research also with collaboration with a Swedish group, which is a Swedish Research Council. So I thank all of them, thank my researcher, and at the end I thank you for having me here, and thank you for your introduction, for your patience. I'll be happy to take up questions and comments if it is not too late for lunch. Thank you very much.
Thank you, sir. Thank you very much. It was a very nice talk to listen. I think our students, our undergraduate students, have benefited highly from the undergraduate introduction, and how this kind of chemistry, it helps in understanding biological models to study them. Okay, if there is any question, I did not find anyone in the chat box. I think sir will be happy to answer. Is there any question from anyone?
Can I ask a question to sir?
Yes. Many. You are not hungry.
Sir, Dr. Shilpa Banerjee of Bethune College, Department of Chemistry. I want to ask you a question that is not a question. I want a suggestion from you. Can we make a token experiment in our UG lab to enhance the interest of biomimetics towards the students?
Oh yes.
Is there any suggestion?
There are many. I think the most exciting experiment that once, in fact, used to, you can do a very simple experiment like binding of oxygen, not to iron, because they are quite oxygen sensitive compounds. So maybe the infrastructural facility at the undergraduate colleges are not sufficient to synthesize very air sensitive compounds. But I would recommend that you can go for the cobalt saline compound, and you can show them how reversibly it can bind oxygen. It's a more like a reversible oxygen binding mimic for hemoglobin. And that's actually not a very difficult synthesis. It's actually your undergraduate students can do this synthesis on their own. You have to just take ethylene diamine and salicylaldehyde and then make a condensation sheet-based product, and then you prepare the cobalt-2 compound, bubble oxygen through it in solution. You will see beautiful color change, and then again you bubble nitrogen gas through it, and you can actually see that the color is gone. If you have a spectrophotometer, I think that would be even better. And it would actually excite students. In fact, I have a lot of module. Because I love teaching. So I have a lot of module for such kind of experiments. So if you are interested, you can write me, or we can discuss over phone. And
yes, sir, I am very much interested because I know you very much. I have done my PhD from ISS. He was there.
Oh, okay.
I was a scholar of Professor Shashwati Lahiri in 2003 to 2010. Dr. Shilpa Banerjee now I am.
I see. Oh, now I know you.
Yes, sir. So I am very much interested to do some typical interesting reactions for the students. And send if you don't mind, then you can. I will give my email ID, and if you give some
absolutely
procedure to me, so that I can enhance the interest among the students, it will be very helpful.
Yes. You know that we have a group comprising of Professor Uday Moitro and some other colleagues.
Exactly. Yes, sir. I know that.
We go to different colleges and try to. Right now, due to the pandemic situation, things are really difficult. But let us all pray and wish that things will be better soon, and then we can have a kind of such workshop where I can show such kind of hands-on experiment to the students also. I mean, other than really helping with some kind of an experimental procedure or some methods, some module,
Yes,
so that
sir,
you
yes,
can
sir.
Yes,
And you told that you have make a book chapter actually. So
yeah.
this book chapter, the procedure, I think this will be helpful for us actually. Is it?
But this book chapter is purely the research results of
Okay, advanced level.
research results. So this is actually, you know, a comprehensive coordination chemistry, which is published in every 10 years. And normally the experts from all over the world, they contribute here. So there are two chapters out of, I think, 50 chapters. Two chapters are contributed by authors from India, and both are from ISES. Myself and my colleague Abhishek, we have two chapters. I have the non-heme part. I have the non-heme chapter, and Abhishek has the heme chapter. So that's something like a Mahabharata for coordination chemists. So yeah, so it's a more like a research result. If you like, I can send you the PDF file so that you can browse through sometime at your free.
Okay, sir, I will contact you to get the procedure, and thank you for nice talk you have given here. Thank you, sir.
Thank you very much.
Okay. So now may I request Dr. Gautam Bandyopadhyay, HOD Department of Chemistry. Sir, can you please end this session with your vote of thanks?
Thank you all. Good morning, good afternoon, everybody. Respected Principal Madam, honorable speakers, our respected DBT Coordinator Dr. Amita Kaur, our most valued speakers, our beloved departmental colleagues, staff, and ladies and gentlemen. It's my privilege to have been asked to propose a vote of thanks at the end of the webinar. I, on the Department of Chemistry and DBT Star College Chemistry Chapter, extend a very hearty vote of thanks to our honorable beloved speakers, Professor Govindo Sami Mugesh, an inorganic physical chemist, a professor and head of the Mugesh Laboratory attached to the Department of Inorganic and Physical Chemistry at the Indian Institute of Science, and Professor Tapon Kanti Pine of Indian Association for the Cultivation of Science, Professor of Inorganic Chemistry. An area of research is inorganic chemistry and biomimetics. I must mention our deep sense of appreciation for their explanation, giving excellent coverage of their subject, exposing their ideas to encourage us and our students. Therefore, we are all inspired by their great deliberations. Again, I would like to take this opportunity to place on record our heartfelt thanks to DBT, Department of Biotechnology, for the perfect logistic support and guidance, and choose our college as a star college of DBT. I also extend my thanks to our mentor, Principal Professor Krishna Rai, for her necessary help and cooperation to organize the event. Next, I express my thanks to our friend, philosopher guide, Dr. Amita Kaur, convener of DBT, Star College, Bethune College chapter, and officiating principal, whose continuous help, cooperation, and support makes the webinar a grand success. Again, I extend my heartful thanks to my departmental colleague, Dr. Paramita Majumdar, Departmental Coordinator of DBT, Star College, for her hard labor, well-managed anchoring and conducting the webinar. Again, I express my sincere thanks to all my departmental colleagues, staff, students of my department, and also my college, whose virtual presence and participation makes the webinar a grand success. I also express my special thanks to Mr. Akash Mondal for his technical support to the smooth running of the successful June session of the webinar. Lastly, I express my heartfelt thanks to all sections of people who help us to make the webinar a grand success. With this, I stop here. Thank you all. Namaskar.
Thank you, Gautam Dutt. Thank you very much. All good experiences come to an end, and this wonderful webinar, these two very nice lectures, very beneficial for us as well as for our students. They have also ended, but I hope that we can get help from Sir Professor Pine, Professor Mugesh, for further experiences for our students. If we approach them for such small demonstrative work and these things, workshops, if we approach them, we will further get help from them. With this hope, we end this webinar here today, and there are feedback link, and for participants, please fill up your feedback link, and you will get your e-certificates in due time. Thank you all for participating. Thank you.
আচ্ছা পারমিতা, স্টুডেন্টরা চলে গেলে আমরা একটু কথা বলে নেব 2 মিনিট।
পরে এখান দিয়ে বেরিয়ে।
হ্যাঁ।